infectious disease, time is absolutely everything. So today, we're going to explore a couple of tools that are completely revolutionizing how labs identify bacteria and fungi. We're talking about 16S rRNA and ITS sequencing. And believe me, this isn't just a small step forward, it's a massive leap into the high-tech future of diagnostics.
Okay, so imagine this. You're a doctor, and you've got a patient with a really serious infection. But the lab results, they're taking days. Or even worse, the organism is just so unusual that it's not showing up clearly under the microscope. In those situations, every single minute counts. This is the exact problem where those classic traditional methods start to, fall short. For decades, and I mean decades, the gold standard was growing microbes in a petri dish. We call it culturing. And don't get me wrong, it's still incredibly useful. But what happens when the bacteria are just too picky to grow? Or maybe the fungus is so rare, its shape doesn't even match what's in the textbooks. Yeah, when life is on the line, just waiting around isn't really an option.
And this is where modern science gives us an incredible breakthrough. We now have a way to get answers that's not only faster, but way more precise. It's a method that lets us completely bypass that slow process of culturing and go straight to the source, right to the microbe's genetic code. So what's the solution? It's called molecular sequencing. And here's an easy way to think about it. Instead of trying to judge a book by its cover, what a microbe looks like or how it acts, we're literally opening it up and reading its unique genetic ID card to find out exactly what it is. At its very core, molecular sequencing is basically like a universal barcode scanner for life. Every single organism has unique genetic sequences, and by reading a very specific part of that code, we can identify it with just incredible accuracy. It's a total game changer for both speed and precision. Now, in the world of clinical microbiology, two of these genetic barcodes have become absolutely essential. For bacteria, we use a marker called 16S rRNA, and for fungi, we turn to a different one called ITS. Let's dig into how each of these works. All right, first up is our bacteria detective 16S rRNA. This is the specialized tool for unmasking bacterial infections, no matter how good they are at hiding. So how does it actually work? Well, that rRNA part stands for ribosomal RNA, which is a key part of a cell's protein-making factory. 16S sequencing looks at this specific gene because, get this, all bacteria have it, but it has tiny little variations between different species. This makes it perfect for identifying those rare or fastidious organisms. The really picky eaters that just refuse to grow in a lab. It just cuts through all the confusion when other tests fail and helps doctors choose the right antibiotic right from the get-go.
To put it simply, when the lab runs into some mysterious bacterium that just won't be identified by normal methods, 16S sequencing is the detective that comes in and cracks the case.
Okay, so if 16S is our bacteria detective, who do you call for fungi? Well, that requires a totally different kind of specialist. So let's meet our second tool, ITS. ITS stands for Internal Transcribed Spacer. It's a section of fungal DNA that's kind of sandwiched between essential genes. You can think of it as genetic padding. Now, this padding isn't super critical for survival, so it mutates and changes much faster, which creates a lot of variety between species. This is absolutely perfect for telling different fungi apart, whether we're trying to distinguish a common yeast from a dangerous invasive mold. And for patients with compromised immune systems, getting that ID fast can literally be life-saving. And that's its real superpower. When a fungus is just impossible to identify by how it looks, its morphology, ITS slices right through the confusion and gives a clear, definitive genetic answer. So, the key takeaway here is this really simple but crucial distinction. These are two highly specialized tools in the diagnostic toolkit. 16S, that's your go-to for bacteria. ITS, that's the precision instrument for fungi. They use the same genetic principle, sure, but they're aimed at completely different culprits. Okay, so we have these amazing, sophisticated tools in the lab. But how does reading a bunch of genetic letters actually translate into better healthcare for a real person? Because at the end of the day, that's what truly matters. Well, the impact is just enormous. Getting a faster, more accurate ID means so much less guesswork. It drastically cuts down the chances of misidentification, and that ensures patients get the right treatment right away. This kind of targeted therapy is especially critical in complex cases where a patient's life might actually be on the line.
It really creates this direct pathway to better care. See, it all starts with getting that precise ID from 16S or ITS. That knowledge then empowers doctors to pick the most effective targeted therapy, which so often is the key to solving some of the most difficult and dangerous infectious disease puzzles out there.
This leaves us with a really fascinating question. We've already gone from waiting days for a culture to getting a precise genetic ID in just a matter of hours. So, as this technology gets even faster and cheaper and more widespread, what comes next? How is it going to reshape our entire approach to fighting infectious diseases? That, right there, is the exciting frontier we're all heading towards.